So, tēnā koutou katoa. Hello, everybody. So my name is Michael Bartlett. I work at Scion in Rotorua in New Zealand in the Pathogen Ecology and Control team. I'd like to start by acknowledging the Gadigal people of this country and elders past, present and emerging. I'd also like to acknowledge ngā hapu e toru, or the three hapu that are mana whenua for the land upon which the Scion facility resides, where this research was conducted. So it was really nice actually being part of quite a big team that worked on this project that brought together expertise not only in pathology but also in using the technology that's required to capture this kind of hyperspectral and thermal data, and then being able to analyse that data and interpret the patterns from it. Cool. I'd also, I think I'll start with a few more acknowledgements as well before I get into the actual talk. So acknowledging that the funding for this work came from Scion, from the SSIF funding pool. We're able to do this work because we have roseapple or Syzygium jambos in the Scion nursery, and Auckland Council provided that material quite a few years ago now. There were several people involved that helped with disease assessments and looking after the plants while they were part of while the project was happening. And then the Scion nursery staff for propagation and care of plants, because without those that work we wouldn't actually be able to do any of this. This project has actually, there's a manuscript that's been accepted for publication, and you can get an early access version that's available online at the moment. So if you're interested in more detail, then please go and look for it online. Cool. So we all know what myrtle rust is, and we're probably pretty aware that it can be quite problematic in nurseries, right? So nursery conditions can exacerbate the issue by creating microclimates that are favourable for disease infection and spread amongst plants, and it can be really challenging to control myrtle rust in a nursery setting. In New Zealand, at least, it's an offence against the Biosecurity Act to move infected material. But nurseries don't want to ship infected plants out the door for a number of reasons that are pretty self-explanatory. And we know that in New Zealand, nurseries use a couple of different methodologies or tools primarily to control myrtle rust. And those two things are fungicides. So Rob Beresford and colleagues have done a lot of work showing that fungicides can be used really effectively to control myrtle rust in a nursery setting. But fungicides can't always be used, and some nurseries don't want to use fungicides as well, so it's worth noting. And then the other primary methodology is surveillance and removal, so getting staff to look through the crops if they see disease symptoms, then removing that material from the nursery. I want everybody to now place themselves into the mind of a nursery manager. And you are in charge of this consignment of crops that are sitting at the bottom photo there, so quite a lot of seedlings sitting there on that table. And one of your staff has come up to you and said, oh, we found myrtle rust on these plants. And it's not on many of them, it's on a few, and it's kind of scattered throughout the table. And so your first port of call is to obviously remove the infected plants. They're due to go out soon, and you have to make a call as to whether you think it's okay to send that material out into the environment where it might be infected, but the symptoms have not yet to express, so we've got asymptomatic infected plants. And how do you go about figuring out whether that's okay? You can't rely on conventional diagnosis because there aren't any symptoms, so that's out the window. You could potentially use DNA-based techniques like qPCR, and we've heard a bit about the qPCR already, but where do you sample, right? Really difficult. You kind of need symptoms to guide sampling in that case. And so this was actually the scenario that our nursery at Scion was placed in last year. And talking to the nursery management team at the time, one of the things that they asked for was whether there was a way to scan plants and detect whether they were safe to ship or not. And of course, we don't actually have that technology at the moment, but talking to the team at Scion who work in this space, if I can get it to go. There's potential for hyperspectral and thermal imagery to provide information about whether plants are infected. And we've already heard talks in the conference giving the overview that when pathogens infect plants, there's a physiological response in the plants. And this kind of data can actually detect quite subtle changes in plant physiology. So the objective here was to really investigate whether this would be feasible as a first look. And to do that, we inoculated rose apple plants under quite controlled conditions using these tents that we built in our containment lab. We had a total of 81 plants to work with, so pretty small sample sizes, and we were quite constrained by space. Plants were actually put into these conditions which were conducive to symptom development for a period of time before the experiment, and one or two of them were removed that developed pustules, so we were pretty confident that the plants we were working with were clean to start with. And we've kind of heard a bit about rose apple and why it's such a good model to work with, so I won't spend time talking about that. So the sort of design of the experiment was that measurements were made before inoculation took place. Then there's a little bit of a gap where plants are in the dark and under conditions that are ideal for spore germination, so you kind of leave them alone. And then following that, measurements were taken every day for 6 days, so from day 2 to day 7. And then we did another data capture at day 10, but we didn't actually end up needing to use that data. So I did my job well. Symptoms developed on all of the inoculated plants. And no symptoms developed on any of the control plants, which was a bit of a relief. And we actually continued to monitor for symptom development up to day 17 on that control group, just to make sure that we were confident there was no infection there. With the inoculated plants, we knew the day in which the visual symptoms first appeared, so we could reclassify those plants in terms of 3, 2, or 1 days before symptoms appeared, the day that symptoms appeared, and then the days after symptoms appeared. What measurements were made? A very close visual inspection was made of all the new flush material on both sides of the leaves, and we actually used a hand lens for this. And when we talk about the day that visual symptoms appeared, the top picture of that leaf there is kind of what we're talking about, and you might not even be able to see it. Very, very subtle, tiny little red spots, maybe some little bumps appearing on the leaf surface, and we only really know that that's actually myrtle rust because we followed and tracked symptom development through until pustules appeared. So in the nursery setting, you wouldn't have any clue that that's actually infected at that time point, at least anyway. Thermal data was collected using a camera above the plants and with standards of known sort of temperature. And then hyperspectral data was collected using a spectroradiometer, taking point measurements from both the new flush leaves that are affected by myrtle rust or directly infected, as well as the mature leaves on the plant which aren't directly infected by the pathogen. Data was analysed and models were developed using 101 different hyperspectral indices for data from both the green leaves or mature leaves and the new leaves, as well as thermal indices across the whole canopy. But that's a lot of predictors in your model. Anyone has any idea about statistics, you don't really want to be doing that, especially with only 81 plants. So recursive feature elimination was conducted to reduce that to a more manageable level and really pull out those features or indices that were most informative. And then classification was undertaken using regularized discriminant analysis. That's a machine learning technique, and the team actually looked at random forest and another method as well, and this turned out to be the best. And I know that with these machine learning type techniques, you normally have a test data set and then a full data set. We had 2 small sample sizes for that, so they used a 5-fold cross-validation type method to work for this. So the results that come out, and I don't want to spend too long on this table because there's lots of acronyms there, which doesn't really tell you much, but we do see sort of patterns that come through from these models. So the best models had indices that showed up repeatedly throughout, and for hyperspectral data collected from green leaves or mature leaves, that ratio 975 and the healthy index were pretty common in the models. For the new flush leaves, you can see the other 2 there, they were common across models. And then 2 thermal indices, the normalized canopy temperature and the standard deviation of normalized canopy temperature were used for the thermal models. Now, the results are pretty interesting. The thermal indices turned out to be very good at classifying whether plants were infected with myrtle rust or not from 1 day before visual symptom development. with perfect allocation into the two groups. And hyperspectral indices were also quite good at certain time points, and you can see that the F1 scores and accuracy are pretty high and considered really good at several different stages there. So what does this look like when we look at the data? So we've got the thermal image data at the top there, and you can see that the two groups diverge quite nicely. The hyperspectral indices are a little bit harder to interpret. There is a pretty clear trend at the bottom from data collected from the new flush leaves, where we have a reduction in those values in the treatment group compared to the control. So talking a little bit more about the thermal data and what that kind of might mean for what's going on inside the plant. And this is another way of visualizing that data. If you plot the two different indices against one another, you can see that they all overlap. And then once you get to 1 day before symptoms, there's a pretty clear divergence between the two groups. A reduction in canopy temperature, we think, is associated with infection by myrtle rust due to increased transpiration rates in the leaves caused by damage. And this is something that's been noted in other biotrophic pathogens like rust and mildews. And then that standard deviation in canopy temperature is also increased because of variable infection throughout the canopy compared to the control plants. So in conclusion, as a proof of concept, we had indices that could separate plants out, and they also give us some insight into maybe the physiological processes happening in the leaves. Indices from susceptible red leaves, for example, are associated with reductions in chlorophyll content. Thermal indices were very, very good, surprisingly good, and definitely worth further investigation. And these results suggest that an accurate characterization technology could be developed for use in nurseries in the future. But we acknowledge that the sample sizes for this work were quite small, so we need to do more work. And we need to be looking at how this might work on different host species, because rose apple is only one of many. So thank you very much.
Thanks, Michael. A really interesting talk. I'm wondering if you've still got a sampling problem, even with your thermal imaging. So how can you scan a whole consignment of plants before they go out, do you think?
Yeah, well, that's the challenge, isn't it? It's developing a tool that's actually usable, and something that a lot of analysis goes into interpreting that data and making it usable. So we're at early stages. This is sort of proof of concept level.
Sorry. Oh, sorry. I'm just wondering how you control for a similar response caused by different pathogens.
Yeah, that's a really good question as well. Changes in responses to plants outside of these heavily controlled conditions could also, really complicate the picture. And so the next step will be definitely to look at other pathogens, other things that can induce stress like drought and so on, to get a better picture of that. Cool.
Really nice talk, thank you. What do you think the prospects are for scaling up the thermal and the hyperspectral imaging to airborne systems?
Yeah, well, I know that this kind of technology is used from drones for other types of questions. So there's potential there to try and collect thermal or hyperspectral data from an unmanned aerial vehicle over forest canopies. Again, this was very controlled. A nursery setting is slightly less controlled. Going out into a forest, even more so. And so there's potential there, but I'm not sure how well it would perform.